Hi everyone, I'm Karen McRae-Johns and I'm the Marketing Manager at Millennium Science and Germa Life Research. Together we are Australia and New Zealand's leading distributor of scientific reagents and instruments for biomedical research. Firstly, I would like to thank all researchers for your efforts to continue your important studies during these unprecedented times over the last couple of years. We've faced lockdowns, border closures, poor government funding, and for some limited access to the lab and office. In today's webinar, a few of my colleagues and a special guest speaker from TEDX Genomics will be providing short snapshots about the latest COVID-related research tools and products that might be of interest to you. If you would like to learn more about what's going to be discussed today, please fill out our poll that will pop up on your screen after the last short talk, or contact us via email. We should have a few minutes after the last presentation for a few questions. So firstly, I would like to introduce Saba Fanagi, our product manager for our antibody and protein portfolio, and she'll discuss COVID research tools from Sino Biological. Over to you, Saba.
Sure. Let me share my screen. You're all seeing my screen? Fantastic. Thank you so much for the introduction, Karen. Today we are quickly going through the update on COVID-19 products, specifically antibody and proteins. So when you're thinking about the COVID research, what we are thinking about, what are the best source of COVID research for proteins and antibodies? Although there are many products, even from our different supplier, today I would like to highlight Sino Biological product for COVID research. The reason is that Sino was the first company that produced COVID-19 reagent, and their catalog is very large and growing every day and have very quality products. Sino indeed is our 5-star supplier. They have a strong package of relevant validation data. Site operation passes inspection by JOMR. We have no or low history of quality issues with their product. The performance of the products are guaranteed, and they have rapid delivery, especially during this last challenging years. As I mentioned, they have great package of validation data. For example, we are looking at S1 RBD validation data. As you see on the top panel, the purity is more than 95% confirmed by SDS page and HPLC. And at the bottom panel, we are looking at the functional activity of ACE2 binding, which was performed by Biocore and ELISA. Basically, most of their protein products have purity of more than 95%. They come in format of lyophilized. They have high stability, very low endotoxin level, and they're coming in the different pack sizes. Typically, proteins 100 microgram and antibodies 100 microliter. If you're interested in bulk packages, they're offering that with very special pricing. Sino is a key reagent bank for SARS-CoV-2 research. The reason is that they have more than 290 recombinant SARS-CoV-2, more than 200 different SARS-CoV-2 variants, and more than 20 biorelated antigens. And they've got 16 antigens of SARS-CoV-2. Also, they have more than 100 antibodies for SARS-CoV-2, including neutralizing antibodies, specific antibodies, and monoclonal paired match antibody for antigen detection. Plus, they also have the COVID detection kits in format of inhibitor screening kit or antibody titer assay kits. As you all know, there are different emerging variants of COVID over the last few months, and fortunately, Sino Biological covered all of these variants. These are including the most concerning variant, Alpha, Delta, Beta, and most recently, Omicron. Now, Sino Biological put the Omicron production, and I received lots of questions from our researchers and customers asking what is the best source of Omicron and who has the Omicron. So here you go, Sino has it. And they have a very comprehensive range of Omicron proteins now available. You are looking at the different Omicron variants that are available by Sino. 5 out of these 8 variants with the different antigens and tags are now available to order, and 3 of them on these 3 are ready for pre-order, and hopefully they are coming to their catalog by end of this month. Also want to touch base on the antibody against Omicron. As you know, probably Omicron spike region has more than 30 mutations in that region. That's why it makes it concerning, because researchers thinking that is the cause of more transmissibility of this variant. And it's very important to see if this more than 30 mutation allow this variant to escape from neutralizing antibody. And that's very important to know for vaccine development or drug discovery. So Sino screened all of their developed neutralizing antibody against this variant. And interestingly, only one of their clone that you see at the top with this catalog number has very high reactivity with Omicron. This is the antibody that was developed last year and now has showed very high reactivity with Omicron, which might be of your interest to test against any of the Omicron protein that suits your study. So I finish up here and just let you know that we are offering the best price and fastest delivery on Sino biological products. Sino was very successful in developing a very big stock of all of their proteins and antibodies and serving lots of researchers and biopharmaceutical company in Australia and New Zealand. And they continue to do that. So if you're interested in any of their products, please feel free to contact us on our general info email address, and definitely I'm more than happy to assist you further with your questions or any inquiries about the details of the products. Thank you for your attention. I stop sharing now and pass on to my colleague.
Okay, so next up is Paul Gooding, who's our field application scientist for our genomics portfolio, and he'll be discussing PacBio's new HiFi viral product. Over to you, Paul.
Thanks, Karen. Okay, hopefully we can see this. Minimize myself, which is always a bit. Okay, so just introducing the PacBio HiFi viral SARS-CoV-2 kit here today. So why this kit? Well, up to now, mostly short read next-gen sequencing has been used to sequence the COVID genome, and that's relied on like a raft of PCR primer panels in a sort of massive multiplex in order to amplify short targets that essentially tessellate across the about 30 kp viral genome. So this type of panel takes a lot of time, obviously, to optimize in terms of the primer designs themselves, getting even coverage, balancing the TMs of each primer, optimizing primer concentration so you don't get dropouts and all that sort of thing. So you do all that work, and then of course a new variant comes along with mutations, as we know. So if a mutation occurs over one of those primer sites, of which there's thousands of them, then you can get dropouts in the coverage, and we see those cases where effectively you get a sequence with a piece missing, and you can use that incomplete genome, if you like, to infer lineage of the viral variants. But if you don't sequence everything right, you don't know what you might be missing, and that can be really important. So there's a couple of examples here comparing the Delta variant that we all know about against the brand new Omicron variant that's come across, and this is just the spike protein region here. So this is just the spike protein piece, and you can see all the cluster of mutations that sit here. I've circled the ones that actually fall on a primer site in red here, so you can see that it causes real problems. You can see that this this without any mutations that are between those primers in the amplicons themselves, but just on the primers, you may not get proper binding. You may get complete dropouts of those sequences. Okay. Now, the primer sets have to be continually sort of updated, reassessed, and that can be a real pain. I'm just an example here of a couple of the commercial sets, the Arctic primer sets, which are used for Illumina sequencing, the short read sequencing, and also the Midnight primer sets, which are used by ONT, the noisier long read sequencing. So it's a real burden to have to update these, and as you can see, we're already on the third update of the Arctic primer set since June. So you know, you take the time to order the primers, you have to optimize all the new multiplexing, make further changes to the primer design perhaps, and then get the relative concentrations right so you get the even coverage, change the PCR cycling conditions, and all of that to get a robust test. And you're doing this you know, onwards and onwards as you get new mutations, which is a real pain. And of course, there'll be new variants coming where we're just going to have to do this. So the PacBio HiFi viral kit is essentially there to give labs the chance to stop this sort of panic stations redesign of the assays. And the approach that they've used, I think you'll see as we go to the next slide, is another example of their kind of game-changing technology, and it has fundamental advantages in providing the most complete, contiguous, and accurate genome information for all existing variants, and with no need to update the panels as these variants come along. So how does it work? Well, this is the PacBio solution. They use molecular inversion probes, or MIPS, rather than a standard PCR sort of assay. So the molecular inversion probes, it combines like the tiling capability of a target capture assay, but with the simplicity and low cost of a sort of PCR assay. So you've got these kind of backbones here, these sort of semicircles, and then they have the probes on the end, which are homologous to the SARS-CoV-2 genome. And inside you have a section which is essentially the viral target piece that you're going to sequence. And this is paneled right across the genome, 1,000 probes. You get at least 22-fold coverage of each base along the entire genome. And this is why it's very resistant to a lot of these mutations, because you've got so much coverage there. And it's also very tolerant of samples where the quality is not quite so good. And obviously with the two probe arms, it's very, very highly specific target as well. So as we can see here, if you take the existing SARS-CoV reference at the top against the new Omicron variant, you see you get this kind of 22-fold coverage across the entire genome here, except in the spike protein where all the mutations exist for Omicron. But even then, we still end up with a minimum of 10 probes at each position. So it really is giving you confidence that you're going to get good sequence data from here. So in the kit, well, everything's included. There's no need to source anything from third-party providers or vendors. It's the complete workflow. It gives very high-quality results over a very wide dynamic range of samples. As I said, not susceptible to problems with a lot of sampling with low quality, which is a real problem with PCR-based assays. It's a very flexible kit. You can scale from 24 samples up to 384 samples at a time, and the entire workflow can be run from sample prep right through to results in about 24 hours on a Sequel II or 2E system. Importantly, the hands-on time is kept to a real minimum, and there's as few touch points, as it were, as possible to avoid problems with errors due to contamination or just handling errors. In fact, it's really neat, I thought, in the kit. Each reagent that you add changes the color of the solution in the wells. So you can see when you've added each reagent in the order, and you can see if any wells have been missed or anything like that by the color change. I thought that was actually pretty neat. So a lot more convenient really than an amplicon-based system. In terms, again, of the workflow, if you can automate it, of course, but if you're doing it manually, you start with 6 microliters of extracted RNA from your sample, goes into an overnight cDNA prep and hybridization. Next day is the enrichment steps, followed by a PCR, and then the pooling of all your samples. And then the second day would be a library construction, and then loading on the instrument. You walk away, and next morning you're going to have results. Okay, and what's very neat about that is that on a Sequel 2, using their SmartLink 10.2 software, it launches automatically through the pipeline to provide consensus sequences for each sample and a report with coverage and QC information. It even flags samples that might contain multiple COVID variants in that single sample. So just some information for ordering if you're interested. The HiFi viral kit, part numbers at the top there. The other pieces underneath are the bits you need for your library prep and sequencing on a Sequel 2 on their SmartCell 8M. If you need more information, please contact Millennium Science. The details are there, or contact me. I'm Paul, or you can contact my colleague James at the information there. So with that, I may have run over a minute or so, so I apologise, but thanks very much. I will hand back to Karen. Thanks.
Thanks, Paul. Next up, we have a short presentation from Jeff McDermott from TEDx Genomics, and he's a science and technology advisor. So over to you, Jeff.
Thanks, Karen. So at TEDx Dynamics, we have a pretty simple yet very lofty mission, and that is to build products to interrogate, understand, and ultimately master biology. Specifically, our products combine hardware, chemistry, and software to give single-cell and spatial views of biological systems. And whilst there are numerous key application areas that technologies are used in, I guess for the purpose of today's session, I really centered the presentation around new tools for SARS-CoV-2 research. And here's a nice infographic which really outlines how our products map onto the key infectious disease applications. And these range from monitoring immune response, elucidating fundamental cellular mechanisms, right through to therapeutic discovery. And that's what I really want to drill down on with this brief 5-minute presentation. And really trying to answer the key question being, how can single-cell immune profiling kit be used for antibody discovery, particularly within the framework of studying SARS-CoV-2? So we're extending the capabilities of our single-cell immune profiling kit to enable what we call our BEAM, or BEAM-AB in this case, application, which is a paradigm shift in the way you think about antibody discovery. BEAM stands for barcode-enabled antigen mapping, and put simply, it allows you to map full-length VDJ sequences from B-cell surface receptors to their cognate antigen specificities. Right now, you can use our single-cell immune profiling kit to survey and catalog full-length paired VDJ sequences from B cells. On their own, the surface receptor information is really powerful, but to fully understand the immune response, we also need the ability to map the antigens that these receptors recognize at scale. So if you like, we're currently assessing half the problem, and then BEAM will enable you to examine the other half, or really see everything as a whole. Here's a high-level overview of the antibody discovery workflow using BEAM. And again, put it simply, BEAM allows you to map B-cell surface receptors to their cognate antigen specificities. So to walk through this, firstly, your barcode antigens of interest, the DNA oligo, and then apply them to a population of B cells. Next, B cells that have bound the antigens are isolated through either FACS or MAX-based sorting, and then this enriched population of cells is simply loaded onto the Chromium system. By tagging multiple antigens with barcodes and staining B cells with these antigens, you can index single or multi-antigen specificity back to thousands of single cells and their corresponding heavy and light chains. 10x software will then help identify B cell receptors of interest for downstream testing. So basically, to this workflow, you supply the biotinylated antigen, and we supply everything else. As an example, we used BEAM antibody at the start of this year to search for SARS-CoV-2 neutralizing antibodies in the blood of patients infected with COVID. As you can see, we started with PBMCs, exposed them to 5 different SARS-CoV-2 antigens. They had all been barcoded separately. Then we sorted for antigen-bound B cells and then ran them through the Chromium instrument. So ultimately, from 100 million PBMCs that we started with, we identified about 2,500 B cells specific to the 5 SARS-CoV-2 antigens that we exposed those 100 million PBMCs to. This workflow took less than a week, which makes it a really powerful tool for high throughput antibody discovery. To highlight just how significant that statement on the last slide is, here is a contrast between BEAM and a typical hybridoma workflow that would currently be used for antibody discovery. Without diving into too much detail here, you can see that the hybridoma-based approach to antibody discovery takes significantly longer timeframes and can actually return fewer clones. Furthermore, with our BEAM solution, researchers can isolate cross-reactive antibodies against multiple strains, and it can be used as a complementary solution to interrogating previously hard to access lymph nodes, so things like bone marrow. So on this last slide, let's jump back into that large SARS-CoV-2 antigen data set that we just saw on a couple of slides and take a little bit of a deeper dive into that data. So first of all, keep in mind that B-cell receptors can bind more than one antigen because of the variable region maturation. So the epitope that is recognized by the B-cell surface receptor may have common binding to multiple antigens, basically. The 5 barcoded SARS-CoV-2 antigens mapped in this patient's serum are shown in the horizontal bar chart. And then if the isolated B-cell contains sequencing reads for a given antigen, the circle is colored black, and the vertical bar chart itself is just the number of times this B-cell configuration is counted. So this graph is really important because it shows how broadly neutralizing the antibody is. And this is not based off convoluted bioinformatic algorithms from sequencing bulk B-cell DNA. These are physical counts with real associated bound antigens. And really, before this product, this is something that's unprecedented. So from this analysis, we identified that most antibodies are broadly neutralizing and recognize all current variants of concern with picomolar affinity. And remember, we're able to obtain this information from a single experiment that took about a week from sample through to analysis, and truly game-changing stuff if you're really interested in antibody discovery. So thank you so much for your time today. If you'd like to learn more about our products or about how that BEAM solution works or how you can implement it in your lab to accelerate your antibody discovery process, then please just reach out to me. My details are on the slide here, or we can answer some things in question time. Thank you.
Thanks, Jet. And to wrap up our Bitesized Bioproduct briefing today, I'm going to pass over to Anthony Baghdadi, who's a product manager for our protein analysis portfolio. So over to you, Anthony.
Cool. Hopefully you guys can see my screen. So today I'm just going to talk briefly about surface plasmon resonance, or SPR, with just a couple of examples of how open SPR from Nicoya can be used in COVID research. And it jumped. So SPR has really been the gold standard for measuring molecular interactions for almost 2 decades now. And it's essentially a label-free technique that's used to measure binding kinetics for a wide variety of molecules. So what type of data can you actually expect from an SPR experiment? So imagine you have a big library of molecules and you want to see whether any of those biomolecules can actually bind to a specific target. So you can answer the question, does it bind or doesn't it? How specific that binding actually is, how strong the interaction is, so the affinity, how quickly 2 biomolecules can actually bind, which is the on rate, and how quickly 2 biomolecules can come apart or dissociate, which is the off rate. So the main benefits of SPR are that no labeling is required, so you don't need any fluorescent labels. Experiments can actually be done on samples in their native state. You don't need to use a lot of your sample. So for those who work in protein purification or with crude samples, you know how little yield you can get from those sorts of techniques and how precious your samples are. So you get real-time binding kinetics. So you don't have to wait a couple of days or a couple of weeks. You can get that binding information on the fly as you're actually performing the experiment. And you get a lot of flexibility with the applications you can do. So you can look at protein-protein interactions, protein-antibody interactions, protein-small molecule interactions, etc., and so forth. So Nicoya's open SPR technology is a little bit different from the traditional SPR methods that use a thin layer of gold. So open SPR actually uses a nanoplasmonic biosensor, which is essentially just nanostructured gold particles. So in this case, what we do is we measure a change in the absorbance peak instead of a change in angle, which is what traditional SPR instruments do. So here the gold nanoparticles would absorb light and create a distinct absorbance peak for the gold. And then as binding events occur on the actual sensor, you're going to see a shift in the absorbance peak, which is translated to a response in a sensorgram, as shown on the right. So the advantages to this system is that there's actually minimal background interference because the system works with localized SPR fields. You also can get much higher sensitivity because you're working so close to the sensor surface. And the optics are much more simple and robust. You just have one LED light source that shines onto the sensor and through to the detector on the other side. Traditional SPR is a little bit more complicated because you're looking at reflected light, and the positioning of the light source and the detectors needs to be at appropriate distances for sensitivity. So because of this simplified optics, the instrument actually is a lot smaller than a traditional SPR instrument. So it's actually a benchtop type instrument. So all this really culminates in a really more in a more affordable system that's a lot cheaper to maintain. So I'm just going to take you through 2 quick examples of how OpenSPR can actually be used in COVID research. So in this study published earlier this year in Nature Communications, they were looking at the trimeric spike protein of human coronavirus 229E, which binds to human aminopeptidase N, or HAPN. So in this study, 2 different conformational states of these trimeric spike proteins were identified by cryo-EM, and they were found to actually so binding of HAPN was found to be favored in one particular conformation. So OpenSPR here was actually used to show that one of these conformations, they called the activated state, has a much higher binding affinity compared to the closed state. And so this is because the receptor binding domain is actually exposed in this conformation. So it's really important to be able to understand how binding of these trimeric spike proteins can actually be affected by the conformation of these proteins. That's really important for the development of COVID therapeutics. So another example here is actually an application note from Nicoya. So what they did was they used varying concentrations of serum ranging from 150 nanomolar to 1.85 nanomolar. And they basically screened this serum to look at the binding kinetics of the SARS-CoV-2 antibodies present in the serum. And you can see from the graph on the left that you get very strong binding kinetics across all the concentration. And what was done here as well was the secondary antibody was used to demonstrate the potential to improve the limits of detection of the system. And this actually increases sensitivity quite substantially, giving sensitivity down to picomolar concentration. So that means that the system can actually be used to detect for binding events, even for very dilute samples. So in this way, you could theoretically also use OpenSPR as a diagnostic tool to look for the presence of viral-specific antibodies, for example, to determine the effectiveness of a vaccine. So all in all, OpenSPR can be used to investigate real-time, highly sensitive binding kinetics for a wide variety of molecules in a small, user-friendly, low-maintenance system. And from the present examples, you can see that it can also be a really useful tool in COVID research. That's all from me. Thanks for your attention.